Our next presentation is Evotec, and I'd like to welcome Andrea Schiel, Executive Vice President, Head of Cell Therapy, to the stage.
Good morning, everyone. It's a real pleasure to be here. Thanks for actually attending and coming to listen to my presentation. It's fairly early in the day, so it's great to see quite a few people here. It's also a great opportunity for me to actually present on what's going on in our company, in our efforts on cell therapy. So we'll dive right in. So I'll be discussing a little bit the projects that we're working on, the platform that we've built. I'll cover a couple of topics that are and have been challenges in the allogeneic iPSC-based field, and then I'll talk a little bit about a flagship project that we have and that we really care about. So I don't know, you may not know us as a company. We're actually quite a sizable company now. We've grown quite significantly over the last decade or so. We're more than 4,000 scientists working across all modalities, discovery, development, manufacturing, providing services in many areas. But as you can also see on the top right-hand side, we also pursue alternative business model where we make internal R&D investments to create specific and unique platforms, to create early project starting points that we then preclinically seek partnerships for to drive them into the clinic. And that business model is certainly something that we use in the cell therapy space quite extensively. So just looking, in what we do, so we believe the future of cell therapy is allogeneic, and in particular, we believe it's iPSC-based. Now, that may not be true for all indications, but for many indications where patient numbers are large, like in autoimmunity, or in the regenerative space where access to cells in an autologous setting is impossible, whether it's diabetes, whether it's CNS, whether it's heart regeneration. And so we feel iPSC-based therapies just offer so many benefits, and that's outlined on that slide. I mean, I'm aware this is an expert audience, so I'll go through that quite quickly so you can see, you just require from the top going clockwise, you require material from a single donor. You make a GMP line, a GMP bank, and that bank is a major asset. You can essentially derive all kinds of cell type from a single bank. Further down on the right, you can see the gene editing can be done very precisely. It's then at the iPSC level. You can make knock-ins, knockouts, you can combine them. And then further at the bottom, one of the main assets is going to be a clonal gene editor MCV with the gene edits of choice, and then a single vial can be thawed and actually expanded and scaled up to the required level and to manufacture drug product and to end up with an off-the-shelf a frozen drug product. And such a gene-edited MCV can actually literally last for the entire lifetime of a commercial product. Now, it's fair to say this sounds great on paper, and it is actually great, but technically this is tremendously challenging. And so when we started out in this area, we realized that several years ago, lots of those capabilities were actually not available in the market, so we decided to build them actually in-house. So we have built what we call an end-to-end platform, going from the idea, what about therapeutic cell type X, up to actually a clinical manufacturing, and you can see all the different disciplines and areas that we've built in-house. We have a bit more than 100 people purely working on iPSC-based cell therapeutics, and that's really a core expertise is iPSC biology in the company. And one core element, you can see that on the right, is, as we heard from many speakers in the previous days, is manufacturing and moving projects into CMC. And we felt that is something also that we needed to build internally, so we acquired a manufacturing site in Italy that we largely use for iPSC-based therapeutics. As I mentioned earlier, our business model is beyond building this end-to-end platform, is to invest in early projects. You can see the different shades this takes. If you look at the bottom, there are some areas where we purely build GMP-compliant protocols, starting with the GMP line to make multiple cell types. And you can see that at the bottom. In the middle part, you can see that we also focus on specific projects that we drive forward, and we currently focus on diabetes and oncology, increasingly also in autoimmunity. And you can also see that at the top, we have already been successful in striking a number of partnerships to help move those projects to the clinic. We have a partnership with a pharmaceutical company in the oncology space in gamma delta T cells. We have a partnership, and I'll talk a little bit about that later, with Sanofi in diabetes. And a couple of weeks ago, we announced a partnership with Novo to really work on actually process development and make manufacturing procedures ready for clinical and also late-stage clinical or even commercial use. So for us, this has been a fantastic validation of the efforts that we've put in and the platforms that we've built. So moving to a couple of the challenges and how we deal with them. You know, for those in the field and working with stem cells, you will be aware that genetic integrity in the stem cell field is a challenge, and you know, a whole range of companies have burned their fingers with this, and at least we believe this needs more attention, and it also needs to be discussed in more detail. So yes, the statement that, you know, stem cells can be expanded indefinitely, that is true, but in reality it's actually not true, because these cells have a propensity to pick up genetic aberrations. So if you go to a high passage number, you actually can't use them for therapeutic products anymore. And so each passage, you can see that on the cartoon, if you have if you culture them and you start to expand them, you have a mutation in a gene that provides a survival or growth advantage. There is the danger that cells carrying this mutation will actually be enriched and eventually take over. This is, of course, a bit excessively shown on the cartoon, but this is something that really needs to be looked at, needs to be taken care of. This is often caused by cellular causing stress on the cells at the iPSC stage. So the solution is to be really careful what conditions you choose for GMP-compliant conditions. But more importantly, it's really to establish a very comprehensive set of analytical assays that allows you to actually monitor this in the lifetime of the manufacturing procedure. And this is shown on this slide here. You won't be able to read all the details. I think that doesn't matter. On the table on the top right, you can see some of those methods are completely unbiased, and they go from karyotyping at a very high level to whole genome sequencing. Unfortunately, those methods are not incredibly sensitive, so if you have only a small population of aberrant cells, you won't be able to see them, which is why we use more hypothesis-driven methods on oncogenes, on DNA repair genes, through digital PCR and other methods. And the key element in this is also, from a regulatory perspective, especially if the cells are gene-edited, at least the regulators in the EU are fairly rigid on the procedures and the processes they want, and we've discussed that with them, and they're happy with the panels of characterization analytical assays that we're actually using. One additional areas, and you know, time is too short to really go into the scientific details, but for allogeneic therapies, of course, the rejection of transplanted or infused cells is a key challenge. And there's a whole range of strategies through gene editing that allows to block different immune pathways, and many of those are described in the literature. I don't want to talk about them today, but we've actually now spent a few years to develop proprietary methods, and there's lots of actually learnings. Initially, we thought we're going to develop a platform, it's going to work for all kinds of cell types. Well, that's actually not quite true. Actually, every cell type needs its own tailored set, and especially once you move towards GMP compliance and how is this executed under GMP, it becomes really complicated. In a normal lab, everyone can do gene editing, but doing this under GMP with as few passages as possible, maintaining genetic integrity is a real challenge, and it's taken us years to actually develop that procedure, and we're just about to move the first gene editing project into CMC. The last part is about showing some data on some projects that we're working on, and diabetes is an area that we've cared, and we have a long history in diabetes R&D, and the field in type 1 diabetes has been longing for decades and dreaming about this vision of curing the disease through regenerative medicine. And while we're still not close to that, we're moving closer every year, and this slide just shows our own efforts. So we can now make iPSC-derived, what we call islet-like clusters. You can see that on the left-hand side, some images, and we have developed a manufacturing procedure that is GMP compatible, starts with a GMP line to manufacture those islet-like clusters. And this is probably the most complicated manufacturing procedure under the sun. You're making little organoids that, of course, have beta cells, because these are the cells that sense glucose and secrete insulin. But you need other cell types in there as well, alpha cells, delta cells. There are other cell types you certainly do not want in there. So, making a drug product that's efficacious but also safe is a big challenge, where we can freeze the cells. And we're at the stage implementing this, in our GMP facility. And on the right-hand side, you can see some preclinical data. This is mouse data. And you can see on the y-axis, I don't know whether you can read the y-axis, this is actually glucose level. So at the beginning, you made the mice diabetic by destroying the pancreas, and then to transplant the cells in the mice. And you can see it takes a while until the cells kick in. But then eventually, through a single dose, the mice are essentially cured. I don't know whether you can read the x-axis. So in this experiment, we've looked for 1.5 years, and the cells just get better and better, and there is no indication that they will decline in terms of function. And so this is quite remarkable. We can compare those data to donor islets. We can predict clinical doses. We use clinical biomarkers to assess this. So I think this is really exciting. This is really what gets us out of bed. If you look at the blue curve, the mice actually arrive at the human set point of glucose sensing. So their pancreatic system is actually humanized through the transplant, and this is really, really remarkable. Moving to the clinic, though, is just another story. You may have heard Jonathan's presentation from Sanova yesterday, I believe. And so we've actually teamed up. So Sanova have this really cool medical device called Cell Pouch that is pre-implanted. It gets vascularized, which is for beta cells really, really important. They need to sense glucose. They secrete insulin, but they especially require lots of oxygen. And if that's not provided, the cells will just degenerate quite quickly. There is clinical data using that Cell Pouch that Jonathan actually presented yesterday, using donor islets, so we know that the device actually works in people, and it keeps cells alive. And we feel this is really cool because it makes the transplant retrievable, and it also overcomes some other challenges that are present in device-less transplantations. So we've been working together for a couple of years, and on the right-hand side you can see similar experiment than what I showed just a minute ago, but it's now cells in the pouch, again transplanted in mice, and the efficacy is literally the same. It's a bit more noisy in this experiment. We've looked for up to 3 months, but again there is no indication that this will actually that the function will actually go down. So now we have a clinically validated medical device for transplantation. We have iPSC-derived islet-like clusters that we know are equipotent to donor-derived islets. We know they're actually more robust than donor-derived islets. We have preclinical data that demonstrate that the cells work in the pouch, and now the path is to really implement this under CMC and to test this into patients. And we're tremendously excited about this. And I just want to end up with a final slide on, what this means for the diabetes area. This is only the start. The initial strategy will require immunosuppression, and this is, of course, only suitable for a smaller number of patients with hypoglycemic an awareness, but we're already working, and we're moving this currently into CMC on gene-edited cells that we think will be protected from allo rejection. And you can see that there are a number of project opportunities, whether these cells are transplanted in an open device or in a deviceless, remains to be seen. If you're interested in any of those projects, please come and talk to us.
Thank you, Mr. Schiel.
and
We'd like to invite our
that's-
next
I'm
presenter
done.
to the stage.
Thank you.